Okay. So good evening, all the participants in this Science Dialogue series initiative. This is very short course, 5 hours course, but very relevant course. Those who are pursuing their research in molecular biology, genetic engineering, synthetic biology. This is very, very important course. 20 years earlier, Craig Venter designed artificial chromosome, or you can say artificial microorganism they have created, and Craig Venter. So it was like a दावानल की तरह वो टॉपिक पूरा ये हो गया कि आर्टिफिशियल कैसे बना दिया ऑर्गेनिज्म. Still only 0.1% of bioscientists knowing that techniques, है ना. So we have requested Dr. Sanjay Ghosh sir on this topic because he has expertise. I also take, but since participants are less most of the time, so we don't take, and I don't have hands-on training on all these techniques of DNA assembly. So I have invited Sanjay Ghosh sir. I have already introduced him in previous session as well as in this flyer in your Google form also. You all have gone through his expertise. So briefly, as you can see, he is working as associate professor at IBAB, Institute of Bioinformatics and Applied Biotechnology, private institute but very reputed institute situated at Bangalore. And before joining here, he has spent decades in USA, France, and other countries as a postdoc fellow. He has gone through different postdoc fellowship in different top institutions. We have already mentioned his things. And he's also a very good person. He is trying to disseminate knowledge to the society. So free of cost also, he has delivered 2-3 lecture under Science Dialogue series, free lecture. We are in touch with him since several years, at least 2 years. So he is always ready to deliver lecture. So I heartily welcome Dr. Sanjay Ghosh sir in this Science Dialogue series, in this course, from my heart, bottom of my heart. Thank you, sir, for accepting our invitation and taking this long 5 hours courses. Obviously, those who are attending, they will be their knowledge will improve in this synthetic biology stream, artificial, you can say, genome synthesis. I request all the participants to interact as maximum as possible so that you will learn. It's not possible always to go through hands-on training, but theoretically also those who have gone through that experiment, if you will discuss with that mentor, you will get most of the experimental things online also or through slide also. This is my experience because we teach 12-13 types of polyacrylamide gel electrophoresis, 12-13 types of PCR. So we try to give them that experimental things also. So I request you to utilize his expertise. Now, sir, platform is yours, and thank you again for taking this pain, this course. Ultimately, we are here despite of you have injury in this teeth injury, Hannah.
Yeah.
Yes, he is suffering, but I have requested एक week तो already हमने extend कर दिया. So participants को और extend ना करे. So we should start. So he will take 45 minutes or 1 hour today, and then after this Diwali vacation, he will take. But you get glimpses of this topic, how important it is. So, sir, now platform is yours.
Thank you, sir. Thank you for a very generous and kind introduction. So I don't know if I deserve all of that accolades that you gave me. So I must also congratulate you. You are doing a great job, and yeah, more and more participants should come and attend these talks on variety of topics, which Gopalji sir does, as you all know, online, free of cost. So I really appreciate your efforts. So this is my small contribution in kind of sharing the knowledge that I have. As teachers, we always will like to, share the knowledge that we have, and for the next generation to pick up and also, take it forward, innovate, etc. So before I start, I will just like to say that as Gopalji sir mentioned, please interact. You can, stop me at any slide or anything that you don't understand. I can also speak Hindi, so हिंदी में बात कर सकते हैं. तो आप लोग जरूर मुझे टोकिएगा और मुझे पूछिएगा कि अगर कुछ समझ में नहीं आया. And after that, I mean, I will also go little bit slowly. So today, जो मेरा plan है कि I will start. So there are variety of DNA assembly methods, as you all know, right? So I will just start with the so-called the simplest one and the basic one. But before going into the technical detail, as Gopalji sir mentioned, that these techniques are very important for making artificial or synthetic DNA genomes, right? Gopalji sir mentioned correctly Craig Venter. He was the first one who made an artificial chromosome of mycoplasma. And this was a big thing in those days. And we'll look into those examples later during the course, how it was done. And actually, all of you, if you try and if you have the resources, we can also do it in the lab nowadays, right? So I will just start first with the synthetic biology and why DNA assembly methods are a very important technique for the synthetic biology or even genetic engineering processes. So just for bandwidth, I will just close my video and then I'll just go ahead. Okay, so what are the different main technologies for so-called the next generation biology? So we talk of future of biotechnology, right? And one of the things that we have been doing for genetic engineering for the last almost 25 years was cutting, pasting DNA. And that is most thing that we could do. But for the last around 10-15 years, now there are a lot of technologies have come which are going to impact the area of biology and in principle, so-called synthetic biology or next-gen biology as we call it. So this next-gen biology is possible because of the three main technologies. One is that nowadays we can read DNA, and read DNA means we can sequence the DNA. And sequencing also, as you know, was there from Sanger's time. Fred Sanger got a Nobel Prize for finding a way to sequence. And I'm sure that at least some of you, if not all of you, you know how Sanger sequencing is done. And I'm not going to talk about it. But you also know, I'm very sure, or you have heard about it, something called NGS, which is next generation sequencing, which is the next technology or a superior technology. So Sanger sequencing is called the first generation sequencing technology. And after Sanger, whatever has come from 2000 onwards is called the next generation sequencing technology. And this has allowed basically to sequence massive amounts of DNA in all at once. So it's also called massive parallel sequencing, right? So that means we can read now any gene, any genome of any organism or any cell type more or less quite easily. Within 1 week, we can do that. So any organism, we don't have to clone. We can just isolate the DNA, we can fragment it, and we can put it in these fancy machines. And these machines will basically give you the sequence output. So this is one such machine which is shown here, and these are high-seq, next-seq. There are a variety of seq things, right? So these technologies are around 20-25 years old, and they are allowing us to read the genome of organism for which we have no clue beforehand. So therefore, reading is very important. The next technological innovation that has happened is writing the genome. So now we can make artificial genes using this machine, which is used for gene synthesis. Why I'm saying that we can write? Let's assume that I want to have a new gene, right? I mean, which does not exist in nature. I have engineered the gene. Nowadays with AI and other kind of things, you can design proteins, design genes which do not exist in nature as of now. So how to then test or study their function? So one of the ways is that, as biologists do, is that they will synthesize the gene artificially. because you cannot clone from organism. That organism doesn't exist. So you can send it to a chemical these companies which gives the genes synthesis service, give them the FASTA file which has the sequence of your gene, and they will synthesize for you. That means we have the power as a biologist to now write the genome which doesn't exist. So this is a very important, a key technological innovation that has happened. In addition to writing, and you have all, I'm very sure you all know, we can now edit the DNA. So this is like you are reading a newspaper, or we are having, some kind of reading material. So we can read, we can write on a paper, and we can edit also what is already written. And how do we edit? And these are the three main technologies which were available. Now all of us are aware of CRISPR-Cas9 because of Nobel Prize to Jennifer Doudna and Emmanuel Charpentier. So this is nothing but a CRISPR-Cas9 based method which just generates a double strand break at a place in the genome that you will design, right? So this is as simple as that. Before CRISPR-Cas9 came into picture, the two older technologies which were there before that, which was used to change the sequence of the DNA as per your wish and will, was called zinc finger nucleus, or ZFN in short, and something called TALEN. So these two are DNA binding proteins which are used to create a double strand break, which is shown by a black arrow, and therefore was used to change the sequence of a DNA at a place of your choice. So the take-home message from this slide is very simple, that now we can do synthetic organism, we can design some new organisms, new genes, new properties by having these three key technologies to be able to read, write, and edit what is pre-existing. So writing means completely new, reading is something that is already there, and editing is something which is already there but you want to change as per your wish and will. So why we need, or why these technologies are important? So they are important because the ultimate goal in future is to create new biological system, not necessarily biological organism, but at least a new system. System in the sense, can we use the cell to produce or to behave in a way that we want, right? And to do that, we should be able to engineer the genome. We should be able to put new genes into the genome. We should be able to read what is there in the genome, right? These informations are required. So this is the ultimate goal which the biologists or the next gen biologists, they plan for, and these have using these three key technologies, and this has huge impact. As you might have seen already some examples where artificially created genes, for example, yeast nowadays makes carotenoids, right? So the carotenoids that we get tablet or capsule is not now made from natural sources. It is made in yeast. But normally the yeast generally doesn't have genes to make carotene. So then how does the yeast make it? The yeast makes it because people have put genes from other organism or completely new genes into yeast, and the yeast can now used as a biomanufacturing factory. So there are a variety of uses of these three technologies. This is just mentioned here in terms of medicine. You can now cure diseases, and there are some examples for that, although not in clinic as of now. And also for environmental monitoring, that if there is a pollutant, if there is a toxin, etc., we can create and design new organisms and new biological systems which can detect that and warn us that this water is polluted or there is a contaminant in the soil, etc., and etc., right? So these are the three technologies I will like you to keep in mind that is driving the so-called next gen biology. Okay, so since I work in the area of synthetic biology, and Gopalji sir also mentioned that these DNA assembly techniques are central to work in the area of synthetic biology. But the first question comes, how do we define synthetic biology? So synthetic biology, there is no clear-cut definition, but what scientists kind of give a loose definition is that it involves basically designing and creating or constructing new biological parts. So that means, as I gave the example of yeast, yeast doesn't make carotenoids, but can you now design new genes and promoters, etc., devices and systems which can make a product which yeast naturally doesn't make? Or we can redesign. That means I can take an yeast cell, I can just change some of the genes of its own and make it do something that naturally doesn't do. So that is also synthetic biology, right? Because naturally yeast doesn't do that, but I have changed the genome in some way so that it does something that I want. So either we can put something completely new or we can redesign. And for redesigning, we need these DNA assembly methods. So synthetic biology, as you all know, most likely it is nothing but engineering the biological system or a cell. And this is to basically, the end goal is that it should have these cells or this biological system should give an output or a function which do not exist in nature before. So again, yeast doesn't make carotenoids. I engineered it to make carotenoids, right? And this also should be having a human benefit. So not just any genetic engineering, right? So in nutshell, synthetic biology is nothing but a marriage between the biology as well as engineering principle. And that's a separate topic that I don't want to go more into that. But there are some talks I have given earlier. You can go to YouTube and you can see that what are the examples of synthetic biology and how an organism can be engineered. What are the different examples? So here, broadly speaking, the principles are the same as an engineering, making a new car, for example, a mechanical or electrical engineering. So in a typical engineering pipeline, we use different parts like nuts and bolts and other things. We assemble a device, let's say engine, etc. And then this entire device, we put it in a aluminum casing, right, which we call as chassis. And that creates a system, because this system allows us to go from a point A to point B. So that has a function. So same way, the synthetic biology also takes, in this case, the parts are biological parts, not mechanical parts. So here we have genes, we have promoters, terminators, regulators, etc., right? So those different genetic parts can be assembled together to make a device. That means it will give a given output. If it is made inside a cell, it will give a novel product, right? And these devices are then transformed into E. coli, transfected into a different host cell, so that the host cell now makes something. So the input can be simple molecules like sugar, but the output will be something complex and something that is beneficial to humans, like medicines, like biosensors, like any other high value metabolite. Nowadays, you might know that, for example, insulin that we use, it made recombinantly from a yeast. Yeast naturally doesn't make insulin. Insulin humans make. So you have basically engineered an yeast which will just, you give the right medium and it will secrete those kind of products. So this is having a function, and the function is novel because that doesn't exist before in the yeast at all, right? So that's the example. Now, what is the workflow? How do people engineer these organisms for a desired output? So of course, there has to be a circuit, and the circuit is still work in progress. In electrical engineers or mechanical engineers, they work on these kind of circuits, right? Where there is a on-off switch, there is different kind of toggle switches and all. The idea is very simple. If you give an input, there has to be an output. Input in biology system should be simple items, and output can be something that we desire. Now to do that in practice, how do we do it in the lab? So in the lab, we have to design first, just like engineers. So you have to design which promoter, which gene, what is this gene doing? So all this information has to be there beforehand. So synthetic biology is nothing but it is applying the knowledge that is already there to create new systems, right? So the first thing comes is design. And then we have to build. That means we have to clone. We have to stitch together these DNA fragments into a new device, right? And that device, this is where the wet lab part comes. That means you will go to the lab and do it. Designing, you can use computers or even simple pen and paper to know from the literature what kind of gene, what is the function, how it is regulated. All this information comes in the design stage. And then you go to the lab and then you order these oligos, PCR and other kind of cloning reagents, and you build it. And once you build it, then you have to test. Is it working? Because you are putting a heterologous system into a cell or an organism which naturally doesn't have it. So you don't know it will work or not, right? So you have to test it. And for testing, after testing, this may work, this may not work. If it works, great. Then you will try to see how I can improve it so that it makes more carotenes, for example. Or if it fails, then we say, fine, can we learn something for it? And then maybe we should change the promoter. Maybe we should add a different terminator. And that is the learning part. And then from when we learn, we go back to the drawing board, design, build, test. So this is a kind of a circular process in synthetic biology. This is called DBTL cycle in short. Design, build, test, and learn, right? So this is a core workflow for synthetic biology experiments. Now, if we look at what are the technologies available. So for design, as I said, there can be computer-aided design. There are different plasmids available. There are some DNA libraries there. Some nucleic acids are also available, which we can purchase. But the build part, where you actually have to go to the lab and learn and have some molecular biology experience, is DNA assembly mostly. So you will have Golden Gate and the different technologies that we will discuss over the next few days. These all are needed to make this recombinant plasmid or a new device, right? We also have, as I said, the gene synthesis. That means we can order a new gene which we don't want to clone. If we have resources, we can ask a company to do it, but that gene we have to clone. So that means this part is still important, right? And now we also will like to edit. I want to make a gene and I want to make it better. I want to introduce some mutations. So I can use different technologies like CRISPR, base editing, and so on to modify the sequence once I have cloned. And then, of course, there are different processes like pooled oligosynthesis. This is mostly used for screening to know that which one of them is the right plasmid or which one of them has the right function. And once we have this cloning done, then to test, we put it in an organism. And we ask that whether this organism is giving the product that we are interested in based on the design and the assembly. So if there is any mistake in design or if there is any mistake in cloning, then this will not work. And that is what we have to learn. That is it because of a technical error or it is because there is not enough knowledge available how the genes will work, right? Because it can fail still. And there, all the data that comes out from the test can be used nowadays to use in artificial intelligence or machine learning to give those data points, and the machine can probably tell us, okay, maybe you should optimize this condition or that condition, or you should put one more gene or two more promoters, etc. Right? So there are some design rules coming out of it, and because of these design rules, we go back to design, build, test, and so on. So I hope the main point here for me to show this slide is to appreciate that this is a very important part, build. And to build, what is we are going to discuss are the technologies which are required or essential to build a new genome, build a new gene, and build a gene with a new function, right? Okay, so now these are some things I will just give an introduction sort of thing that you probably to bring all of you on the same plane, that you have already done it or you already know it, right? So the key tools and technologies is to do a recombinant DNA technology. So what we do there, we take a foreign DNA and we have some restriction enzymes. We digest the plasmid or the cloning vector with the enzyme, a given restriction enzyme, and the genomic DNA or foreign DNA with the same restriction enzyme, right? Both have then, for example, sticky ends in this case. You make a recombinant plasmid which has a selection marker, and that is important because why the selection marker is needed? Because you need to know after you put it in an E. coli, which E. coli has received the plasmid. If you are not able to do, then there is no way of knowing which cell has received, which has not. And why this is important? Because no technology or method is 100%. So that means even if you transform, you give a heat shock, most of your cells would not have received the plasmid, only a few. And to know which few, you need this so-called selection markers. So then you plate it, and then you can have a different kind of selections. Either it is antibiotic based, in this case, let's say ampicillin resistance gene. That means in the plate, you will add ampicillin. Any cell that has not received this plasmid will die because you have added an antibiotic and there is no way these cells will survive. However, if the cell E. coli has received such plasmids, then that will survive. And the other way of knowing that if it has received the recombinant plasmid, but maybe it has not received the foreign DNA, this vector itself can self-ligate because you are cutting with one enzyme. So to know whether it is a self-ligated vector or a recombinant vector, people use blue-white color selection, which you might have done in your practical classes, which basically tell that if it is white, you are disrupting the gene, If it is blue, then you have not disrupted. And if you have not disrupted, that means it is empty vector. The vector doesn't have your insert. So the cloning has failed. But if you have white colonies, that means you have a recombinant plasmid, and that is what you are looking for. So if you have done this experiment, you will see that depending on the ligation difficulty, you will have mostly blue, maybe a few white, right? So usually it depends on what is the frequency of ligation. But the other thing also you might know and you might have done is PCR. And this is one of the main drivers of genetic engineering or even recombinant DNA technology, which is also used for DNA assembly methods, which I'll come later. So these PCR, just to remind all of you, has variety of components. So we need a Taq polymerase, which is thermostable. That means usually proteins denature at high temperature, but not this enzyme. This enzyme rather works at high temperature. So that is good because the PCR process involves three main steps. One is denaturing. That means you have to destroy the hydrogen bond of the double strand DNA by just simply heating, right? So the double strand bonds are broken because these are non-covalent bonds. Once you denature it, then you have to put something called these primers, which are nothing but short DNA oligonucleotides. Short in the sense somewhere between 18 to up to 30, 40 nucleotides. They are complementary to one of the strands, and that is what we have to design. And once you have these primers annealed, and there are usually one pair, two different primers. One is called a forward, one is called a reverse, right? So these two will bind and these will extend. So there will be a 3' hydroxyl group here, and the DNA polymerase will extend. Simply extend both of them. And this is like DNA replication. You copy the template strand, right? And this is all about PCR that we have to know. That PCR is nothing but you need three different steps. You need primers, and these are commercially available. You can buy it. So what is variable is the DNA samples that you put, and what is variable is your primer, because primer will depend on the DNA sample. The rest remains the same in all PCR reactions, right? So this is the take-home message. Now, till now, and you must be aware of that cloning and DNA recombinant, this technology depends heavily on restriction enzymes. That means, and you know what is restriction enzyme, that they are DNA endonucleases, right? And they can create either a sticky end or a blunt end. Now, these are mostly used for genetic engineering, and how they are used, that you again, as I showed earlier, you have some in the plasmid, you have a restriction site, recognition site. So the enzyme will sit and cut, and it will also cut randomly in your genomic DNA. You will basically put them together in a single tube, add DNA ligase, and the ligase will create a phosphodiester bond, and you got your recombinant plasmid. So this is a typical workflow, how it is done. But here you can see the foreign DNA is nothing but a genomic DNA. You want to clone the entire genome of human or whatever cell you want. But important thing to remember, you need to have the restriction enzyme site in the plasmid as well as on the genome. Now, if you don't have the restriction enzyme either in the plasmid or in your lab, then you are in trouble, because then you cannot use the restriction enzyme and you cannot clone directly, and you have to find some other way. And the DNA assembly techniques that we are going to talk about do not depend on restriction enzymes. So that means it is up to you how to do that, right? And we'll discuss that in a moment. So now, not only that you can clone a genomic fragment, but actually you can clone a PCR fragment also, right? So you have amplified a piece of DNA, you have engineered two restriction sites within the oligonucleotides, and that you can cut with the restriction enzymes, and that you can ligate. Or what you can do, you can swap one DNA fragment from one vector into another, provided they both have the same enzyme at the ends of this DNA. So you can digest it, put it together, same way you can digest a plasmid and put this fragment here. So in the end, what you have to do is to take the DNA piece. Origin doesn't matter, whether it is coming from PCR, it is coming from genomic DNA, it is coming from another plasmid. You add DNA ligase and then mix it together, and you hope that it will ligate properly, and you will have enough colonies to screen and get the recombinant DNA after selection, right? Okay, so now I'll quickly go through some of the things of polymerase chain reaction, because most of these methods that we'll talk about is PCR based. So we have to be very clear what is PCR and how it is done and how the primers are designed, because those primer design rules are required for assembling DNA without the restriction enzyme that we are interested in discussing. So PCR, I mean, like you all know that how the nuclear fission chain reaction works. So the polymerase chain reaction, it was named after that because the uranium fission reaction was discovered before that. So here, a given template DNA, it gets copied into 2. So the equation is 2 to the power 2 because if it is perfect doubling each cycle. So 2 to the power 2 is 4. Now this, in the third cycle, it will be 2 to the power 3, 8 copies, 16 copies, 32 copies. So by fourth cycle, starting from 1, you have 32 copies. And typically PCRs are run from 30 to 35 cycles, right? This goes on. So that will mean 2 to the power 36, and that will roughly come to around 68 billion copies of DNA if PCR has worked well. So that means you make a huge amplification, and that is why it is called a chain reaction, because the product of the PCR serves as a template in the next round. These 4 will serve as a template in the next round, and so on and so forth. So it's an exponential amplification. So, but how much you will amplify, what you will amplify will depend on the primer you decide, right? And that is something we'll look in a moment. Okay, so this thing I have already discussed. I'll come to what are the PCR components, how it is assembled, and we'll see how it is important later on. So we will need some kind of template DNA, let's say plasmid, genomic, virus, whatever. The only requirement of a template DNA is that it should contain sequence for this primer binding. Now, what are the primers? Primers are nothing but generally 20 to 30 nucleotides long. They are made chemically. So it is still this day, the primers are made by companies who will make using this gene synthesis sort of chemistry. And this gene synthesis kind of chemistry is still almost the same as Hargobind Khurana had done initially to get the Nobel Prize, almost, I'll say, almost 50 years back, 50, 60 years back, right? So the idea is that you make the small oligonucleotides, and then they should be complementary to the 3' ends of the target DNA. But these primers should not be complementary to each other. And I just want you to all to remember there is something called identical and complementary in DNA. Complementary means that the base sequence is just the matching pair. Identical means same as the template, right? So PCR will have primers which are complementary. They are not identical to the template, right? And they should not contain inverted sequences. Of course, you have enzyme Taq polymerase, which has high processivity and high proofreading activity, right? So what is proofreading and what is processivity? So processivity is that a given Taq polymerase, which is an enzyme, how many nucleotides long it can put in a given time frame. So let's say a given Taq polymerase has a processivity of 1 kb per 15 seconds. So that means in 15 seconds, it can make a 1 kb long DNA fragment. Now you can imagine there is another Taq polymerase which has a processivity of 1 kb 1 minute. So that means it will take 1 minute to put the same amount of nucleotides to make a 1 kb long. So of course, the enzyme which does the same job in less time is having high processivity, right? So we are always looking for high processivity enzymes, not low. And the other thing is proofreading. Proofreading means that it should correct mistakes, right? So this is important because this gives rise to something called fidelity. How similar or identical is the DNA fragment compared to the template? It should be as identical as possible. And to do that, there has been many enzymes that has been discovered, and it is used for DNA assembly. So the original Taq polymerase, which was discovered by Carey Mullis, or which he used to do PCR and got the Nobel Prize, was having very low fidelity. Low fidelity means more mistakes. But nowadays there is an enzyme called platinum superphi 2 enzyme, as the name suggests, is very hi-fi. It is almost 350-fold more accurate than the original Carey Mullis enzyme. So that means this can copy DNA and not make mistakes, right? 350-fold more proofreading activity. So these are the different enzymes. Of course, the more you go towards the top, the more you have to pay for the enzymes. And it is not always possible for all labs to afford these enzymes. So we work, most of the labs will work somewhere in the middle, that they have to manage between proofreading and processivity, right? Okay, so the other thing to remember is that there is a buffer which you might have seen. It just carries a salt and some magnesium. Any enzyme in biological system to work needs magnesium usually. Restriction enzymes need magnesium, Taq polymerase need magnesium, and so on. And of course, to polymerize, you need these DNTPs, and they have to be all the four for DNA: A, T, G, C. They should be high quality and they should be equimolar. That means it should not be only A is more and G is less or something. It should be equal, because most of the times we are copying DNA without knowing what is the exact sequence, right? So we don't want any nucleotide to be underrepresented. So they have to be equimolar, but they have to be in molar excess in the reaction, right? They should be lot than what is needed. Okay, there is another technique called hot start PCR. I will just mention it, but I will not describe it. Hot start PCR is nothing but when the Taq polymerase is, the active site of the Taq polymerase is blocked by an aptamer or an antibody. That means that Taq polymerase will not get activated until unless you put it in a PCR machine. Otherwise, some Taq polymerases, which I mentioned here, the original one, we set the PCR reaction in ice, right? Why? Because we don't want the Taq polymerase to get activated and make something. But there can be mistakes. But hot start allows the PCR reaction to be set up at room temperature. So you don't need to keep in ice, where is my tube gone? And, in setting reaction is ice is sometimes, problematic. So hot start Taq polymerase and hot start PCR doesn't need that. You can do at room temperature on your bench and PCR will be perfectly fine, right? Okay. These are the PCR cycling condition which we will follow when we assemble the DNA as well. So there are, as you all know, there is a template denaturation step, and only this dotted line, whatever it shows, is cycling. This doesn't cycle. This is the initial denaturation, and in the end, there is an extension. But this is the three crucial steps that is allowed for, that allows DNA to be copied into those billions of copies. Okay, so I'll just skip this thing. I'll just basic kind of knowledge about how the DNA is copied. So let's assume that this is a 5' to 3'. This is a double-stranded DNA, as you can see, and this is your template. So what you do is that when you denature it, these hydrogen bonds are broken. Both the strands are broken. So and they are separated. So the 5' is towards the left, but the bottom strand has a 3' to your left, right? So this is called a sense strand, that is called an antisense strand. And now you have designed primers. And this primer, one of them is called a forward primer. So this arrow means this is the 3' end, right? You can see the reverse primer, the arrow is towards the from right to left. So this is the 3' end. So you design the primer so that this binds to the sense strand, this binds to the antisense strand, right? So this is how these primers will go and bind. And these primers in red, as you can see, the sequence is complementary. It is not identical. It is complementary to the strand to which it binds, in this case and in this case. Now, since the 3' ends face each other, the DNA polymerase will simply bind to the 3' end and will simply extend based on the template till the very end. So that is how you will get from a single double-stranded DNA to 2 DNA strands, following the principles of DNA replication. So now, how do you design primers, right? And this is crucial. So primers are always written 5' to 3'. So whenever you write a primer sequence, if you don't mention, it is assumed that towards your left is the 5' and right is the 3', right? So if you are making a reverse primer, which I was just showing before, then you have to reverse it when you're writing in the copy or when you're sending to the company. This is the right orientation. This one you have to reverse it if you want to write 5 to 3, right? So here the 5 to 3' of the reverse primer will read T, C, A, T, and so on, right? Here it is no problem. So this is the first convention. So they should be designed in the correct orientation. The primers also should not be very long, should not be very short, right? So minimum is 18, typically up to 25, can go a little bit high. And then they have a GC content we should try to keep between 40 and 60%, although not always possible, because if your target DNA strand is AT rich or GC rich, then you cannot do anything. You have to design the primer as per your template, right? But what is important is that you can try to keep the 3' end of the primer a C or a G. And what is the 3' end? So this, as you can see here, I will choose the last base of the primer to be either a G, that means it should pair with a C, or with a C which will pair with a G, right? So this is important. Why? Because a GC pair has 3 hydrogen bonds. That means this primer 3' end will be stable. And if you don't do that, if there is a slight temperature fluctuation, AT pair has only 2 hydrogen bonds, it will break, right? So then if this 3' end is not paired properly, no polymerase can extend, right? So detect polymerase cannot extend and your PCR fails. So it is important, one thing to keep in mind, that the primer should end, 3' end should end in C or G. 5' doesn't matter. Then it should also be an exact match. There should not be any extra bases or any kind of less bases. If you are engineering a restriction site, it should be added to the 5', not to the 3' end of the primer. So 3' end is untouchable. I mean, you should not touch the 3' end, and you should have a perfect sequence. Now there are a variety of softwares available online, and these are tools like IDT or Sigma has these oligo ordering pages. It will also tell you if this primers, what you have designed, has any secondary structure. Why this is important? Because if you form a secondary structure of the primer, which is only 25 nucleotides or so, then the 3' end won't be accessible because it will base pair, right, with the 5' end. So you don't want a secondary structure, and these softwares will tell you. If you have, then you have to either change the position of the primer or you have to do something else. And definitely the primers forward and reverse that you will design should not bind to each other. If they bind to each other, then they will form something called primer dimer. And if they form, then this primer is not going to bind to your template. So your PCR may fail or it may become inefficient. So these two are very important considerations when you're designing primers. Nowadays, you will have tools, online software tools, which may design it for you, but you have to know that sometimes the tools are also not perfect. So the tools may make mistakes. You can manually check. These are the design principles that I am mentioning. Okay, so if there are 2 primers that we have to design for every PCR, the primer pair, the TM or the melting temperature, which again a tool will tell you how much is the melting temperature, the difference between the forward and reverse primer typically should not be more than 5. Less the better. More the problem. PCR may fail or it will almost fail, right? So sometimes if we don't design the primer well, even PCR won't work and DNA assembly will never work, right? So this is something to keep in mind. And then when we are using for the primers for mutagenesis, the mismatch bases should be in the middle of the primer, not to the 5' end, not to the 3' end. And finally, you have to know that the primer should not bind anywhere else in the genome other than what you want it to bind. That means you have to check your primer sequence by doing a blast kind of search, that those 25, 30 nucleotides that you have designed should not have off-target effect. They should only bind to the target that you have designed. And there are some tools. So I will ask all of you, and Gopalji sir will share with you this, or actually the talk will be also shared with you. I will ask all of you to go through this website where how there is a nice demonstration how to design a primer step by step. If you have any problem in designing the primer, of any problem in understanding, please let me know in the next session, then I will describe that also in detail, right? So I'm just giving you a small homework, so to say. But this is a very nice article which describes how to do it by yourself. Okay, so now coming back. So cloning, right? Because at the end of the day, we make an artificial gene, we can make a synthetic gene, but at the end of the day, we have to put it in some kind of vector. And this is what we have, I've already described. I'm not going into the detail. What I want to emphasize is that joining DNA fragments is just not done by restriction enzymes anymore. So restriction enzymes, if you have to use ligase because you need to make a phosphodiester bond. But there are many ligase independent also methods, right? And these are also which we will discuss like in the next meetings, Gibson assembly or recombination based assembly. So today I will just quickly kind of describe to you one of the methods which is restriction enzyme free. You can clone, and it is used for DNA assembly, and that is called splicing by overlap PCR. There is also called overlapping PCR. In short, it is called SOE. So S from splicing, O and PCR, right? So the splicing for SOE PCR is a molecular technique which is used very frequently in the lab nowadays to fuse or join 2 or more DNA fragments and also to introduce mutations. How it is done, we'll look in a moment. So the protocol is very simple, right? You amplify fragments by using PCR, and it should have complementary overlapping end. And these primers are special primers. They are called chimeric primers. And I will tell you what these primers are, why they are called chimeric and why they are special. So for now, just understand that you have to amplify two pieces of DNA, which you want to join together, right? Without restriction enzyme, without having any kind of mismatch. So you amplify these two fragments in two different tubes independently. Then you have to run a gel and you will purify those two fragments. After purification, you will do the actual SOE reaction where you will join both of them using PCR, right? No restriction enzyme. So how it is done? What is the experimental protocol? The protocol is first is to design the primer. So you design 4 primers: forward primer A and the reverse primer D. These are the end primers. And then you have something called inner primer B and C. And this B and C have complementary region. So what do I mean by that? Let us say you want to amplify 2 pieces of DNA, right? So this is one piece, this is one piece. And you want to join this right next to it. You don't want anything in the middle. So what do you do? These are the flanking primers. The one which is forward here and the one which is reverse here, colored in red. These are the short oligonucleotides, like 25, 30 as we were discussing earlier. But this is a chimeric primer. Chimera, why? Because the reverse primer B of the first fragment is actually having a complementary region here, but this part of the primer, which is 5' end, is complementary to this part, right? Similarly, the forward primer is half here and half complementary to the other side, right? So this is why it is called chimera, because one part is from one fragment, the other part of the primer is from the next fragment that you want to join, right? Now, since these are to be joined in a head to tail fashion, therefore, the reverse primer of the first fragment will be having complementarity to this part, and the forward primer of the next fragment should have complementary to the previous fragment. If you do a PCR, what will happen? You will get this situation. You will have this part followed by directly this part, right? Which is shown in so one part is a solid black box and the other one is striped. So the striped part will be made here. This is your PCR fragment. If you use this chimera, then you will make a solid black as well as this chimera, right? So that means you need to design to join 2 fragments. You need 4 primers, 2 normal primers, 2 chimera primers with the middle part having complementary regions. That is the first step. So you have to design this on a computer. So there are tools and softwares that will do, but you have to identify the region. Then once you have ordered the primers, they have come and you have 2 different templates. Let's say one is a human DNA, one is a mouse DNA, right? So fragment A is from human, fragment B is from mouse. So then you put a reaction. Now you perform 2 different PCRs with a high fidelity polymerase. High fidelity means high proofreading, the expensive ones, Fusion, Q5 and all. So now you amplify fragment 1, which is nothing but A plus B primer, and fragment 2, which is C plus D. So this is A plus B fragment, this is C plus D fragment, right? Because you want to join both of them, so this middle portion is complementary, right? And you do a typical PCR for each of them. That means to get this fragment, you follow normal PCR. To do this fragment, you again do a normal PCR. So nothing is here out of the box. It's simple PCR. But the primer design is different. Chimeric primers. You have to remember that. Next, what you do, you run a gel, 2 different reactions on a gel on 2 different planes. You purify the products. And then you quantify how much fragments you have got. So you might have done already, or you know that after running a gel, you can cut out the DNA bands of the desired size and you pass it through a kit. So usually this is silica based kit. And these are just following protocols, adding one solution after another. You wash, you elute the DNA, and your DNA is pure. You can also take a PCR reaction and directly put into these columns, right? You don't have to run a gel. So these are basically purification methods. And there you get a pure DNA. Once you get a pure DNA, you have to quantify how much of DNA concentration do I have. So that you need a spectrophotometer, and these are called nanodrops, because you need just 1 microliter of your eluted DNA to know what is the concentration, right? So these are spectrophotometers. Now, once you have find out how much it is, now you do the actual overlap. This name overlap comes from the third step, which is also a second round PCR. What do you do? Now you mix equimolar amount, mole wise, not nanogram wise, right? You calculate moles and then you add equimolar amounts of purified fragment 1 and purified fragment 2. And now you run 5 or 10 cycles without any primer, right? And I will tell you why in a moment. Now this step allows these two fragments to anneal in the middle, forming a full-length template. And then you add the flanking primers. You don't add the middle primers anymore. You add A and D, which was at the end normal primers. You add to this reaction mix and perform some 30-35 cycles more. So that means the total number of cycles is 5 to 10 plus 20 to 30. So you will get around roughly 25 to 40 cycles you have to do. After cycling, then you basically will run a gel and purify. So how does it work? I'll just give you the overview. So this is what we saw earlier, right? And now you have made two different PCR fragments. You join them together in one tube. And now what will happen? These annealing will happen because it is complementary, right? So this part you have made complementary to this. This part is complementary to this. So they will form these kind of so-called dimers. This part is unproductive. Why? Because the 3' end is here. Polymerase doesn't have template to copy. But look at the first fragment. Here the 3' end is here and this is the template. 3' end is here, that is the template, right? So this is a productive one. So in a tube, what you will do, you will add now this outer primer. So this I was telling you have to do a step without the primer to make these kind of possible permutation combination. And then now you do a normal PCR with this outer and this forward and reverse primer. Now what you have done, this will amplify, right? Because this will fill up here, this will fill up here, and amplification will happen because you have put the outermost primers and thus you have joined the DNA. So now you can see that you can actually delete a piece of DNA or you can actually join two different pieces of DNA in a seamless manner. No restriction enzyme needed, just based on a chimeric primer design, right? So this is the simplicity of this method. So in the lab, what we have done, and anyone can do in a way with some technical expertise. So for example, I have 4 different DNA fragments. I will run it on a gel. One is GFP and other 3 genes. I mix all 4 of them in one tube and come with the outermost primers. And I will get if I add all these fragments, I'm expecting a 1.5 kb, I get 1.5. So they join seamlessly. None of this fragment exists. They have all joined together into one fragment, right? So this is how in real life it looks like. So again, coming back to, you can join not just 2 fragments, you can join n number of fragments, but usually it is used to join 4 or 5, right? So this is called extension. So this is the first fragment. You have again designed a chimeric here, and again chimera is going here, right? Each of this junction will have a chimeric primer. And why this yellow? Because this yellow and yellow will be, if they are together, you can make a circle instead of a linear. So if you have primer like this, you will get these junctions. Then you mix this 3 independent PCR products into one tube, you get one long thing, and this you can purify or you can ligate by doing this joining of yellow and yellow, right? So this is how you can basically join multiple fragments without restriction enzyme, without any ligase, just based on PCR and primer design. So this is the last step maybe for today. I mean, I want to describe. We just discussed how to join DNA fragments, but then how this technology can be used to introduce mutation, right? So let's say I want to insert a piece of DNA within this original DNA, right? So in the first round PCR, again, I have to design primers. Since I want to insert this in the middle, this is how it will look like. I have to design primers where these chimeric primers are complementary to this part and this part, right? And I have to amplify this part of this DNA to be inserted, having chimeric primers which are in the middle of this original DNA. So now if I do PCR, I will get a fragment like this, and the middle one will join at the center. In the second round of PCR, by just using the flanking primers, I have inserted my DNA at the place I want based on the primer position. No restriction enzyme needed, no ligase needed. So this is how you can insert a foreign DNA without any ligase or restriction enzyme. You can use it to delete also. I want to delete this part, right? Because of some reason. I will design primers where the chimera has complementary section between these two. That means this part will be missing. Now, if I independently do this PCR and this PCR and put it together, I will get a smaller piece of DNA. And I have deleted the middle part, right? So this is how deletion works. Oh, sorry. And the last one is that I want to introduce mutations without worrying about restriction enzyme. So this is my target DNA. Let's say I want to put a mutation here, X, right? So I will design two primers which have this mutation in the middle. Now I do one PCR using these two, one PCR using these two, mix it together, and I do an SOE PCR. My mutation is exactly where I want it. So that means this primer design is very important. Primers can be used to then introduce mutation, delete a part of DNA, and even add a part of DNA, right? Okay, so I think for today, maybe I'll stop here. We'll continue with more of this application of this technology, and also any questions that you may have, I'm happy to answer.
अब कब लेंगे?
Thursday I can take.
Thursday. So participants, keep in mind 2 days there is no lecture, so he will again take on Thursday. अभी जो है ना होली की छुट्टी है 2 दिन. तो बहुत अच्छा से आपने जो है ना, हम भी ये पढ़ाते हैं एसओई वाला जो है ना, केमिस्ट्री में पढ़ाते थे, लेकिन इमेजिनेशन करना बच्चे के लिए थोड़ा मुश्किल होता है, बट इट्स वेरी इजी टू असेंबल है ना, 2-3 फ्रैगमेंट्स वी कैन ज्वाइन. एनी पार्टिसिपेंट्स हैव एनी क्वेश्चन यू मे आस्क, and during lecture also you can ask question if you have any problem.
Yeah.
Pooja, रितिका, मेरी आवाज जा रही है ना?
यस सर, यस सर.
कुछ है क्वेश्चन पूजा?
सर हाँ, एक वो क्वेश्चन है वो
तो हमको बोलना पड़ेगा. यू ऑल आर फ्री टू आस्क क्वेश्चन एंड इंटरेक्ट, है ना?
नो सर, मैं लास्ट में पूछने वाली थी, अभी पूछने वाली थी.
आज का हो गया और आप थर्सडे को ज्वाइन करिएगा. यू कैन आस्क क्वेश्चन, एनीबॉडी कैन आस्क क्वेश्चन.
ओके सर.
आप पूजा गो अहेड.
सर ये स्पाइसिंग ओवरलैप पीसीआर में जो आपने बोला कि हम वी कैन ऐड मोर देन 2 फ्रेगमेंट्स, तो हमारा अगर मतलब मोर देन 2 और 3 फ्रेगमेंट्स अगर हमारे पास होगा तो उसके मतलब हम ऐड कर सकते हैं ना उसमें, मतलब उसमें कोई प्रॉब्लम आ सकता है?
नहीं, तो मैं जैसे वो दिखा रहा था, आई मीन आई विल गो बैक टू जहां पे ये वाला राइट, ये वाला स्लाइड में जैसे यू कैन सी राइट, अगर 3 फ्रैगमेंट यहाँ दिखाया है, जो जंक्शन है वहां पे आपको काइमेरिक प्राइमर बनाना है बस. तो आप 3, 4, 5 जितना भी फ्रैगमेंट आप ज्वाइन कर सकते हैं, बस ये जंक्शन में आपको ये काइमेरा बनाना है, ताकि इसका 5 प्राइम एंड इसके 3 प्राइम एंड के कॉम्प्लीमेंट्री हो, इसका 3 प्राइम एंड उसके 5 प्राइम के साथ कॉम्प्लीमेंट्री हो. तो बस ये इसका डिजाइन रूल है और कुछ नहीं. एंड अगर ये वो तीनों फ्रैगमेंट को अगर आप एक ट्यूब में मिला देंगे तो वो ज्वाइन कर लेगा ऑटोमेटिक, ये पीसीआर ही कर लेगा.
ओके सर. और सर नेक्स्ट क्वेश्चन ये था कि मतलब ये वाले जो पीसीआर में है उसमें प्राइमर डाइमर का चांसेस ज्यादा होगा एज कंपेयर टू द नॉर्मल पीसीआर?
हाँ, बहुत अच्छा क्वेश्चन, वेरी गुड वन. सो चूंकि ये काफी बड़ा प्राइमर है ना, ये काइमेरिक प्राइमर है, तो इसके चांसेस ज्यादा है. सिर्फ प्राइमर डाइमर से ज्यादा सेकेंडरी स्ट्रक्चर बनने के चांसेस ज्यादा है, क्योंकि हो सकता है कि ये एंड का ये जो सीक्वेंस है ये कुछ स्टेम लूप फॉर्म कर लेगा, तो ये फिर मुश्किल आ जाती है, राइट? तो ये आप जब बना रहे हैं प्राइमर्स, तो ये आपको वो सॉफ्टवेयर जो मैंने बताया, ये आपको बता देगा कि काइमेरिक जो प्राइमर है उसमें कोई सेकेंडरी स्ट्रक्चर है कि नहीं. तो वो ये भी बताएगा कि आप शायद एक जी या सी आगे या पीछे से आप बनवा लो, राइट? सो दैट नाउअडेज देयर आर टूल्स, बट ये एज ए यूजर हम लोगों को ये मालूम होना है कि कहाँ पे चेंज करने से सेकेंडरी स्ट्रक्चर या प्राइमर डाइमर हम लोग अवॉइड कर सकते हैं, ये जानना जरूरी है.
ओके सर, ओके सर, गॉट इट.
आप वो लिंक इसमें चैट में भी दे दीजिए. अंकित पूछ रहे हैं कि प्राइमर एनालाइजर.
हाँ बिल्कुल. तो आई मीन एनीवे मैं आपको अपना स्लाइड भेज दूंगा, आप सब डाल दीजिए, इसमें कोई प्रॉब्लम?
हाँ डाल दीजिएगा, या उसमें व्हाट्सएप ग्रुप में दे दीजिएगा, या यहाँ पर.
हाँ ठीक है. मैं आज ही डाल दूंगा जितना लिंक वगैरह है. कैसे प्राइमर डिजाइन करना है वो भी आप लोग देख लीजिएगा. अगर उसमें कोई क्वेश्चन है तो मुझे बताइएगा.
and you will receive PPT in Google Classroom. I will create Google Classroom. ये Google Meet है, so don't confuse Google Classroom in which I will add PPT.
Sir,
हाँ अंकित, हाँ बताओ.
sir, are we going to cover probes also? Probe designing?
Probe designing?
Yeah.
No, primer designing मतलब.
No.
Probe, Ankit ji, probe means if that is labeled DNA complementary to your target, that is called probe. So
yes, I understand that part, sir. So just wanted to understand, are we going to cover in future those also in future?
No, probe designing देखिए, probe designing in terms of DNA assembly, there are methods to do, but usually we don't do the probe designing. हम लोग क्या करते हैं? We can make this SOE product जहाँ join कर लिए 2-3 fragment, we clone it in a plasmid और उसको sequencing करवा लेते हैं. तो sequencing में मालूम चल जाएगा कि जंक्शन ठीक ठाक जुड़ा है कि नहीं और कोई एक्स्ट्रा बेस है कि नहीं. 99.9% there is no extra base.
Yeah. So no, actually, just wanted to understand for the gene analysis part. When we are
gene analysis के लिए probe चाहिए, but that is like जो आप शायद कह रहे हैं वो शायद qPCR वगैरह का Tachman probe वगैरह ये सब चीज.
Yes, because it's in my work, so that's why I was just.
हाँ नहीं, वो DNA assembly में ठीक आता नहीं है, but वो qPCR के लिए आता है, and that I can mention maybe later on during the course.
Last class में या बीच में आप कभी डिस्कस कर दीजिएगा, है ना? या अंकित जी आप.
Yeah, actually, just wanted to know only the tools which people are using mainly in industries. So those are the major concerns. The links which are majorly being used.
For making probe?
Yeah, for probes as well as the primers, because these are the basic ones. So are there any other best ones? So because I have used those ones, the links you have already sent us.
Okay. No, so what I will suggest ki you can give your requirement, I'll go through it, and then in the next class or in the later on I will give you the links.
Yeah, sure. For sure.
No problem.
Next question है, can we create G block, genomic block?
हाँ, so G block actually gene synthesis में बनाया जाता है usually. So G block बनाने के लिए ये method ही actually is used, SOE, right? जहाँ पे छोटा DNA का fragment we can clone or put it together seamlessly without restriction enzyme. So that's a good question, and it is used by gene synthesis companies to make the artificial genes by designing primers. So I will in the next this course, the next time I will describe a method called templateless PCR, जो SOE की तरह ही है, that is used to make G blocks precisely, but principle is same as SOE. So you are right कि G block बनाने के लिए same principle use होता है.
So Manohar ji, he will discuss G block, and that is G block वही हुआ genomic SOE से भी कर सकते हैं और दूसरे मेथड से भी, है ना?
हाँ.
So you will discuss that, ना?
बिल्कुल.
Yes. Any other question? So thank you for today's session, and I request you all to join on time on Thursday, same time 7 to 8.
Yeah.
Yes.
And from Thursday,
हाँ.
I will do it continuously. मतलब there will be no gap.
Yes. Thursday, Friday, Saturday, and Sunday खत्म.
Yes, Sunday खत्म.
Yes. Practical session means what?
Sorry?
He is asking Manohar ji कि practical session means practical session में क्या आप जानना चाह रहे हो? Practical session hands on तो होगा नहीं. Practical session means he will help you in designing primer.
Yeah. तो मैंने जो लिंक जो मैं भेजूंगा आज, तो आप उसको थोड़ा देख लीजिएगा कि that is practically step by step how to design a forward, reverse, and also that same method can be used to make a chimeric primer. And I will describe when जब हम लोग गिब्सन असेंबली की बात करेंगे तो कैसे ये chimeric primer बनाते हैं वो भी मैं डिस्क्राइब करूंगा अगली बार.
Yes. So thank you and happy Holi to all, including Sanjay sir.
आपको भी और आप सभी को
शुभ रात्रि.
Okay, शुभ रात्रि. Thank you sir.
Thank you, sir.
Thank you, everyone. Thank you.
Thank you, sir.